Hi, this is Dr. Neela Bakore, and we are starting with the first chapter in the unit of reproduction. The first chapter is reproduction in organisms. Reproduction is a process of giving rise to new organisms of the same kind. This is a basic characteristic feature of living organisms. Even viruses, which are kept between living and non-living, they also show this process, a life process, that is reproduction. The basic reason for reproduction is continuity of the race. So why do organisms reproduce? Because they want to continue their race, so that the race continues for times to come. Continuity of the race. This is the basic purpose of reproduction. There are two main categories in which we divide or classify reproductions: asexual reproduction and sexual reproduction. Asexual reproduction is involvement of one parent. So it is also known as uniparental. Whereas in sexual reproduction, two parents, a male and a female, are required. So it is also known as biparental. In asexual reproduction, formation and fusion of gametes is not required or does not take place. So there is no fusion of gametes. Whereas in sexual reproduction, gamete formation and fusion is important part. Formation of gametes takes place by the process of meiosis, and in meiosis genetic variation takes place due to crossing over, whereas in this case, as no gametes are fusing, so no variation is there. So the offsprings which are there, they are identical to the parents. So offsprings are same as parents, or that single parent which is performing this process. Whereas in this, there is genetic variation and the offsprings have characteristics of both the parents, and there is a combination also possible, 50-50, 60-40, or even variable. So genetic variation and the offsprings are different in the sense of the characteristics. So these are basic differences. We will take these processes in detail. Asexual reproduction is also known as apomixis, and sexual reproduction is known as amphimixis. Let us first discuss asexual reproduction and the methods by which this takes place. Asexual reproduction takes place by various methods. So let us first write down the names and then take all these one by one. One is fission. This fission basically is the parent cell splits into daughter cells. It can be binary fission. That means one parent cell splits into two daughter cells, or it could be multiple fission. Another method of asexual reproduction is by budding. the buds normally are termed as an outgrowth on the parent body. So most of the times which we talk of are the buds which are outside. These buds are known as exogenous. So there are two types of buds which are seen. One is exogenous and other is endogenous. So exogenous budding and endogenous. Endogenous budding, where the buds are inside the parent body. Fragmentation. Where the parent body just undergoes fragmentation, breaks up into different fragments, and each fragment gives rise to an individual. Then spore formation. There are various types of spores like zoospores, conidia, oidea, and all those things. And one more, when some vegetative part of the plant is helping in reproduction. So that becomes vegetative propagation. In vegetative propagation, we will talk about both natural as well as artificial. That is, naturally the plants which show vegetative propagation, and artificially how we can help these plants propagate, or we can use these methods for propagation of the plants. So these are the categories in which we divide the asexual reproduction basically. Let us start with fission. So first, fission basically means splitting. One is binary fission. So one type of fission is binary fission, in which one parent cell or organism, it could be unicellular organism, splits into two daughter cells. So one parent cell splits into two daughter cells. Splitting is going to be, first the nucleus is going to split and then cytoplasm will undergo splitting. Now the plane in which this splitting takes place, we can get different types of binary fission, like irregular binary fission. Irregular binary fission. Irregular means there is no fixed plane in which the nucleus or the cytoplasm is going to split. This is seen in amoeba. In amoeba, the cytoplasmic constriction takes place or can take place in any plane. So if this amoeba has to divide or reproduce, first its nucleus will start to show constriction, then this nucleus would completely divide into two nuclei and cytoplasm also starts showing constriction. And now these two daughter amoebae with their nuclei are formed. In this case, we have seen the constriction like this, but because the shape of amoeba is not fixed, it can show constriction in any plane. It can show constriction from a side, from top, from anywhere, and that is why we are calling it irregular binary fission. What has happened to the parent cell? The parent cell undergoes fission to produce two daughter cells. Technically speaking, this cell has now divided into two daughter cells which are again young. They will grow, attain maturity at the normal size and will again divide. So these organisms which show binary fission, they are considered as immortal. immortal because normally what happens is reproduction, then they come into post-reproductive state and then they die. But here when it comes into reproductive phase, it has given rise to two daughter cells. Now both these will grow, again they will divide by binary fission, again two daughter cells will be formed. So naturally they do not undergo death, and that is why they are considered immortal, only the ones which show binary fission. So this is one example where it is irregular. Let us talk about the other type of binary fission. Again binary means parent cell will divide into two, but the plane of the division is decided. It is longitudinal. So longitudinal binary fission. That means it is going to be a vertical type of fission. It is seen in Euglena. Euglena is a flagellate. It has one large flagellum and there is one more smaller which is not visible to us. So when it has to divide, first thing it loses its flagellum. The flagellum is lost, rather both the flagella are lost. And now this upper end, which is called the gullet, Euglena is an organism which is placed between animals and plants. It has plant-like character, that is it has chloroplast, it can perform photosynthesis. Animal-like character is it is without cell wall. Animal cells don't have cell wall. So this has one characteristic of plant and one characteristic of animal. So if it has sufficient light available to it, it will perform photosynthesis. But if light is not available, then it takes food from outside. So its mode of nutrition also becomes heterotrophic. So this is the place from where it would take nourishment or nutrition. So first thing is this upper end, the gullet end divides. So here we start seeing an organism with two gullets formed. The upper end, there are two such structures, and the lower part of the body or cell is still fused. So the splitting of cytoplasm or the constriction is taking place from top to bottom, or it can be termed as vertical. So after this, this constriction deepens. This becomes even deep, and ultimately we would get two daughter Euglena, and now they will develop their flagella. Splitting, or the plane of splitting, is longitudinal or vertical. Again, one parent cell is giving rise to two daughter cells. So that is why it is also binary fission. Because of the plane, it is called longitudinal binary fission. The third type, the plane is transverse. So it is known as transverse binary fission. Transverse binary fission. The plane of cytoplasmic constriction is transverse. It is seen in paramecium. The structure of paramecium, the shape is slipper-like. Here it has a gullet. From here it takes the nourishment. The body is covered with cilia all along. In the gullet also there are cilia. There are two nuclei. Paramecium is a binucleate. It has a large nucleus, macronucleus, and a smaller micronucleus. The macronucleus is responsible for all activities of the cell, and micronucleus is responsible for reproduction. So when it has to reproduce, there are two things which are going to happen simultaneously. the large nucleus will disintegrate. That means it will break up and will dissolve in the cytoplasm. Only the smaller nucleus will remain, and this smaller nucleus will split. So we will see this smaller nucleus splitting into two lobe-like things. Larger nucleus would disintegrate. So this is all disappeared, and only the lower half of the body or lower half of the cell has gullet. That means the food can enter only through here. But if it divides into two, the upper part should also have a structure from where it can take the food in. So here the upper half or upper part also develops a gullet. The lower part anyways has its gullet here. So now the nucleus has undergone splitting, this micronucleus, and the upper and lower parts, they have their individual gullets. The constriction is going to take place from here. It is a transverse plane. So now in the next stage, both these halves, they have their smaller nuclei. Larger nucleus is not yet formed again. Both have their gullets, and the constriction is going to get deeper from here. And finally, these two will separate into two daughter cells. They will develop their macronucleus, all other organelle, and attain the shape of the parent cell. They will grow in size and will become exactly like parent cell. The plane of division is transverse. So it is also called transverse binary fission. In all three, whether it is irregular, longitudinal or transverse, one parent cell is dividing into two daughter cells. So that is why we call them immortal, because there is no natural death. Every time a cell is formed, it is going to give rise to two daughter cells. Now this is fission, the binary one. Let us talk about multiple fission. Here every parent cell is dividing only into two daughter cells. If one parent cell produces many daughter cells, then by splitting obviously, then it will be called multiple fission. So let us take multiple fission now. So after binary fission, let us talk about multiple fission. And after this, we will talk about a slightly different version of binary fission. What happens in multiple fission? One parent cell undergoes fission or splitting to form many daughter cells. And that is why the term multiple fission. It is seen in Plasmodium, the malarial parasite, amoeba, during unfavorable condition. So amoeba shows this, but normally in favorable condition it would show binary fission, but if the conditions are unfavorable, then it will show multiple fission also. Now what happens in this? the parent cell during, or if we are taking the example of amoeba or even plasmodium, the parent cell first undergoes nuclear divisions. So if it is a uninucleate cell, first its nucleus would divide multiple times. Multiple nuclei are formed. So only nuclear division is taking place right now. That is karyokinesis. Each nucleus gets surrounded by little cytoplasm and a membrane. So each nucleus is getting its own cytoplasm and plasma membrane. After this, the parent cell ruptures. When parent cell ruptures, these multiple cells or daughter cells are released. So one parent cell is giving rise to many daughter cells. This is multiple fission. In case of amoeba, what happens is during unfavorable condition in amoeba, it withdraws its pseudopodia, becomes circular and secretes a thick layer around it. This layer is known as cyst, and this process is known as encystment. It remains in this condition throughout the unfavorable condition. So this is during unfavorable conditions. So during that time, it secretes a thick cyst around itself. It becomes circular by absorbing or withdrawing all the pseudopodia, and it has its nucleus as it is. Nothing takes place during this time. So it remains in this cyst form. When the favorable conditions approach, when the conditions get normal, then the cyst is lost. It loses its cyst, its nucleus undergoes division, so it would have many nuclei. All these nuclei will get surrounded by some cytoplasm and plasma membrane. That means daughter cells are formed here, and after this the parent cell will rupture, releasing all these daughter cells. So here one parent cell is giving rise to many daughter cells. It can happen with an encystment stage also, or it could happen straight away like it happens in plasmodium. Now there are some organisms like Opelina and Pilomixa. They show something combination of binary fission and multiple fission. The parent cell, that is Opelina or Pilomixa, which are protozoans, they are multinucleate. Normally, these cells are multinucleate. In all other fissions which we talked of, the cell was uninucleate, whether it was binary fission or even multiple fission. These organisms are normally only multinucleate. So normal condition is multiple nuclei. Now what happens is, this parent cell undergoes binary fission. So when it shows constriction in the cytoplasm, the nuclei get divided into two groups. One group goes into one daughter cell, other group goes into other daughter cell. After this, there would be two daughter cells formed. Let us just count these numbers of nuclei: 1, 2, 3, 4, 5, 6, 7, 8, 9, and let us say 10. Now when the splitting takes place, the distribution of nuclei is not always 50-50. So it is 1, 2, 3, 4, 5, 6 here and 4 here. So the two daughter cells which are formed, they would have unequal number of nuclei, but it is not same as the parent cell. What has happened first? Cytokinesis. In all other divisions we talked of nuclear division first, followed by cytoplasmic division. In this case, cytoplasm divides. Now in both these cells nuclear divisions would take place and they will regain their parent cell size and same number 10 or nuclei in this case, and same here 3, 4, 5, 6, 7, 8, 9 and 10. So the basic difference here also it is fission. It is binary fission only, but the difference is in all other fissions that we talked of, binary, irregular, longitudinal, transverse and even multiple, it is nuclear division first followed by cytoplasmic division. In Opelina and Pilomixa, it is binary fission. One parent cell is giving rise to only two daughter cells, but it is nuclear division or karyokinesis first followed by cytokinesis. So this is how these two organisms are different. So though binary fission but in a different manner. It is, this division is also known as plasmotomy. So slightly different type of division that is seen in these two organisms. So with this we are done with fission. Now let us talk about budding, which is another method of asexual reproduction. So next method is budding. Two types of buds are seen in organisms. Some buds which are seen outside the parent body called exogenous buds, and the ones which grow inside the parent body are endogenous. Exogenous budding is what we have seen in junior classes also. Example is hydra. We also see it in yeast. In yeast, the parent body shows a tiny outgrowth which is known as a bud. This grows to a certain size, and when it attains a size and structure that it is capable of surviving on its own, then it pinches off from the parent body and then it would attach somewhere or free floating. In case of hydra, this is yeast. In hydra, the body is long, slender and vase-like, and they are attached to substratum. They have these tentacles. Again, from the body an outgrowth is seen. The outgrowth is a tiny bud projection kind of a thing. Later on, this will, initially it's going to be just a projection. Then after it attains a size, it starts looking like the parent in every respect, but for the size. So this tiny bud is now going to look like the parent. It has its tentacles and everything. So it's the baby hydra which is growing on the parent body. Once it becomes like of a size where it can survive on its own, it will detach, and this tiny structure will anchor on some substratum and will grow into an adult. We are seeing these projections, these growths outside the parent body. So we are calling them exogenous buds. Endogenous buds are inside. So we don't see it from outside unless and until they come out. The organism where we see these endogenous buds, one is a sponge, that is Spongilla. Spongilla is the only freshwater sponge. and it produces endogenous buds. So, if the body of the sponge has these ostia for inlet of water and the osculum for the outlet, the buds would grow inside here. So, you would not see it from outside till the buds come out. These buds are known as gemmules. These endogenous buds are called gemmules. Now, let us see the structure of these gemmules. In the central part, they have cells which are basically stem cells. They will give rise to the complete organism. These cells are called archaeocytes. These cells, each cell is capable of giving rise to an individual. So, archaeocytes, they are basically stem cells. This complete structure is surrounded by a thick jelly-like mucilaginous substance. The basic purpose of this is to protect these cells from injury or desiccation. At one end, there is an opening. This opening is known as micropyle, and this layer is mucilaginous. In this mucilaginous layer are present spicules. The spicules are needle-like structures. They are of the same diameter in the middle and their ends are pointy like needles. These spicules are known as monoaxonic spicules. So, here in this mucilaginous layer there are these spicules. These spicules are meant for protection. These are pointy structures, needle-like, hard. These spicules can be made up of silica or calcium salts. These are monoaxonic spicules. They can be made up of silica or calcium salts, especially calcium carbonate. Now, when these gemmules or endogenous buds are completely formed, they are released. So, they come out of the osculum, the upper opening. When they come out, they remain in water, free floating structures. Whenever they come in contact with a suitable substratum, because sponges are also attached to the substratum. So, whenever they come in contact with a suitable substratum, they will attach there with their micropyle at the lower end. So, the attachment would be such that the micropyle is here, the cells, the archaeocytes, and the mucilaginous layer around it. And these archaeocytes will come out of this micropyle to form the sponge body. So, the attachment is always through micropyle end. The spicules and everything, then the mucilaginous layer and everything will degenerate. So, the buds, but they are inside the body, called gemmules. The example where we see these gemmules are spongilla. It's a sponge, the only freshwater sponge. All other sponges are marine. So, this is the structure of a gemmule. They are endogenous. So, exogenous outside the body, which can be seen in the form of small outgrowths. endogenous buds are inside the parent body. At a given time there can be many gemmules which are growing in a body, and then all those, when they are formed, they are released from the parent body. So two types of buds. So second method of asexual reproduction, budding is done. Now let us take the next method of asexual reproduction. Okay.